We were studying about what the molecular structure of DNA, and I told it's a polymer made up of what? Nucleotides. Each nucleotide will have one sugar, okay, ribose sugar, one nitrogen base and one phosphate group. So you can see that two nucleotides, okay, two nucleotides. This is another nucleotide, isn't it? See here, you can see a sugar, a nitrogen base and a phosphate. These two nucleotides are joined by phosphodiester bonds, okay, ester bond between two sugar, okay, and you can see that the 5' end will have a free free phosphate group, okay, that is one end of the strand, okay, will have a free phosphate moiety at 5' end and a free OH group, okay, OH group at the 3' end, okay. So you can see that in this 5' ends are in one direction and 5' end or 5' carbons will be in one direction, and in the other strand you can see that 5' end is in the other direction, okay. So this is in one direction, this ribose sugars arrangement, okay, and in this you can see in the other direction. So this is called what? Anti-parallel, okay. So the strands of the DNA are anti-parallel to each other. Anti-parallel means what? The 5' end or 3' end will be in opposite directions in both the strands. Is it clear? The model of DNA, okay, was proposed by Watson and Crick, okay, Watson and Crick, by using the X-ray diffraction data, means the X-ray diffraction data they produced by two other scientists, that is Morris Wilkins and Rosalind Franklin. They have taken the X-ray diffraction of DNA, and from that data and also from the Chargaff's rule. Chargaff's rule says that the ratio between A and T and G and C will be constant and it will be 1, okay. So this is known as what? Chargaff's rule. It was asked once, okay, to write in the board exam. So Chargaff's rules, what it says that ratio between A and T and G and C are constant and it will be equal to 1, okay. So amount of A will be always equal to thymine, and the amount of G, guanosine, will be always equal to cytosine, okay, and likewise A plus G, okay, A plus G will be equal to T plus C. So this is what Chargaff's rule. So what is Chargaff's rule says? It says that ratio between A and T, okay, and G and C will be always constant and it will be 1, okay. So from this only it is found that the amount of A will be always equal to T, G will be equal to C, okay. So from this two, that is X-ray diffraction data, okay, produced by Morris Wilkins and Rosalind Franklin, and Chargaff's rule, okay, Chargaff's rule, only Watson and Crick could propose what? The double helix model of DNA. Understood? And the most important feature of DNA was the strands are complementary to each other, okay. So if you know the sequence of bases in one strand, you can predict the sequence in the other one, okay. So this is known as what? Complementarity, okay. So complementarity of the strands could explain all the process of DNA, like what? The replication, the process of formation of RNA from DNA. So all the properties of DNA could be explained by the double helix model proposed by Watson and Crick, okay. So it could explain all the features of DNA, okay. And so if you find the salient features of double helix model of DNA, it's made up of two polypeptide chains. isn't it? Two polynucleotide chains, okay, and this strand contains what? Sugar and phosphate, okay, and the two chains are anti-parallel, okay. The anti-parallel means if one chain is having polarity in 5-3- direction, the other will have 3-5- direction, okay. So they are anti-parallel each other, okay. That is the carbon atom at 5th position in the sugar component is in one direction, one chain, and it will be in the other direction in the other chain, okay. So they are parallel to each other, but they are opposite in nature, okay. They will be what? Opposite to each other, even though they are parallel, okay. And you can always see that what the adenine will always be seen with thymine, okay, and guanosine with cytosine, okay. So one purine and one pyrimidine, so that the the width or the diameter of the DNA is kept constant. As I told, purines are double ringed structures and pyrimidines are one ringed structures. The pitch, the distance between the pitch, okay, is 3.4 nanometer, and the distance between two adjacent base pair is 0.34 nanometer, okay. The adjacent, see, you can see one CG pair here, one TA pair here, AT pair, okay. So between this two base pairs, the distance is 0.34 nanometer, and likewise 10 base pairs you can see between two adjacent pitch. So this distance will be 3.4 nanometer. Is it clear? Plane, okay, of one base pair stacks over the other in double helix, okay. So this also helps in what? Increasing the stability of the DNA, okay. That is two adjacent base pairs, they exactly stack over the other, okay. One will stack over the other, so that confers or that gives more more strength to the DNA structure, okay. So I told, so this is what the DNA structure proposed by Watson and Crick explains, okay. That is, DNA is a double helical model, it is having two strands, okay. The strands contain sugar and phosphate, okay, and the strands are anti-parallel to each other. Then in between them, what you can see, the nitrogen bases which are joined by hydrogen bonds, okay. The pitch between the adjacent pitch, okay, or the one pitch of DNA is having how many distance? What is the distance between adjacent pitch? That is 3.4 nanometer, and in between two adjacent base pair, the distance is 0.34, okay. So nanometer. So these are what the main features of double helical model of Watson and Crick, okay. So this could explain various what features. I told it explain the complementarity of DNA strands, that is, if you know the base sequence of one strand, the other strand can be predicted. If A is there, then the other strand will have what? T, okay. And if C is there, then it will be G. Likewise, we can predict. Then more than this, the DNA structure proposed by model proposed by Watson and Crick also could explain, okay, the central dogma in molecular biology. That is, the genetic information flows from DNA to RNA and RNA to protein, okay. So this could, that is, this process is known as what? Transcription and translation, okay. That is, DNA, the formation of RNA from DNA is called transcription, and the process of formation of protein from RNA is called translation. So together it is known as central dogma. So central dogma could be explained by the Watson and Crick's model, okay. So how we will explain the length of a DNA? The length of the DNA is usually explained as the number of nucleotides, okay, or how many nucleotide base pair, or you can say pair of nucleotides, okay, so present in it, okay. So for example, human being, Okay, in its haploid content is having 3.3 into 10 raised to the power 9 base pairs. Okay, so that much long length is there in human DNA. Okay, in a haploid set, just imagine this much long, okay, DNA, how it is folded and kept inside the nucleus. So that we will study in the next video, that is the packaging of DNA helix. Okay, so just imagine DNA is very long. Okay, this long DNA molecule and cell, you can imagine, you cannot see cell, then the DNA is found inside the nucleus, isn't it? Inside the nucleus. So how much it should be folded? It should be folded so that it can be accommodated in the nucleus. So this we will be studying in the next video, that is the packaging of DNA helix, how the DNA is packed. Okay, so what is the length of the DNA? How we will explain the length of the DNA? It is explained in the form of what the number of, or it is explained as the number of nucleotides or a pair of nucleotides, okay, present in it. So the human DNA haploid content of human DNA, if you take, that is 3.3 into 10 raised to the power 9 base pair. Okay, so around what it will be, what long. So this long DNA molecule, how it is folded and kept in the nucleus. So that we will be studying in the next video. Okay.